and some of my lab members have spoken already at this conference, SARA, yesterday on DMG. And so I thought I'd mix things up a little bit and talk about an osteosarcoma project. We've been working on this for quite a while, so some of you may have seen some of this data at various stages in different forms as it's evolved. But hopefully I can present a much more complete story, and I think we're getting close to being able to publish this. And of course, as I said, this is on osteosarcoma. So just a bit of introduction for those who don't know much about osteosarcoma. It's the most prevalent primary tumour of bone and the second highest cause of adolescent cancer-related death. And despite quite aggressive standard of care, which includes chemotherapy and surgical resection, the 5-year survival for patients who present with localised disease is really only about 70%. However, for patients that present with metastatic disease or who have disease relapse following or during their treatment, the outcomes are really quite poor, and 5-year survival is really just above 20%. And these survival rates have remained unchanged for decades. And so this really highlights the urgent need for new and improved therapies or strategies to combat this disease. And so, as I said, we became interested in osteosarcoma many years ago now, really based on the fact that it shares these similarities with these primitive bone-forming cells or osteoprogenitors. So it has a high proliferative capacity, it's resistant to apoptosis, and of course it expresses these markers that are consistent with these primitive bone-forming cells or primitive osteoblast markers. And so we've long hypothesised that aberrant activation of key embryonic signalling pathways that are required for normal bone development and differentiation may play a role in the initiation and progression of osteosarcoma. And of course, one of the favourite embryonic signalling pathways in my lab is the hedgehog signalling pathway. So the hedgehog signalling pathway is an evolutionary conserved developmental signalling pathway that's really required for patterning and cell fate specification during development. And aberrant activation of this pathway is associated with quite a large number of cancers. So in mammals, pathway activation is driven by 3 ligands: sonic hedgehog, Indian hedgehog, and desert hedgehog. And in the absence of ligands, this transmembrane receptor patched here inhibits smoothened and prevents its translocation into this cellular organelle called a primary cilia. Now, in this state, this repressive complex, of which SUFU is part of, binds to these full-length Gli proteins and cleaves these into a repressor form which translocates into the nucleus and inhibits transcription of specific target genes. Now, in the presence of ligand, hedgehog binds directly to patched, which alleviates its inhibition of smoothened, and smoothened becomes activated and enters the primary cilia, and it prevents this constitutive processing of these full-length Gli proteins. They are then stabilised into a Gli activator form, which translocate into the nucleus and activate gene transcription. Now, importantly, smoothened is required for all canonical hedgehog pathway activity, and in fact, this is the target for quite a number of small molecule inhibitors that are in various stages of development for cancer therapy. Now, pathway activation in cancer can occur via 2 mechanisms. The first and the most well understood is the ligand-independent signalling, and this is a result of mutations in components of the hedgehog signalling pathway. So, for instance, loss of function mutations in the negative regulator patched, or SUFU is another negative regulator, a gain of function point mutations in smoothened, and amplification of Gli activators. And these all result in constitutive pathway activation. And these are a common feature generally of hedgehog-driven medulloblastoma as well as basal cell carcinomas in adults. And what's interesting is that inhibition of hedgehog signalling tends to be quite effective in patients that have ligand-independent pathway activation. However, the majority of patients in which hedgehog signalling is implicated do not harbour mutations in components of this pathway, and instead they're driven upstream at the level of the ligand. So you can see here that the ligand can be secreted by either the cells themselves or primarily in a lot of cancers by the surrounding stroma. Now, despite the mechanistic and the therapeutic implications of this, there's actually no known biomarker or no reliable biomarker that can predict which cancers that express hedgehog ligands are actually dependent on either autocrine or paracrine ligand-dependent signalling. And so we made an observation several years ago that loss of function events in the tumour suppressor genes, P53 or RB, are commonly associated with ligand-dependent cancers. And this is certainly the case for these adult tumours, GBM, small cell lung cancer, and also osteosarcoma. So we wanted to understand what relationship there might be between P53, RB, and the hedgehog signalling pathway in this disease. And so we started to do this utilising two preclinical models of osteosarcoma. The first is a radiation-induced mouse model using radioactive calcium, CA45. And the second is a P53-RB conditional genetic knockout model in which P53 and RB are specifically inactivated in the osteoblast lineage using OSX-Cre. And so we took cell lines that were derived from the tumours from these mice, and we treated them with exogenous hedgehog ligand and assessed the cells for hedgehog pathway activation. And we did this by measuring GLI1 mRNA. So GLI1 is a downstream transcriptional target of the hedgehog signalling pathway, and so therefore it's indicative of pathway activation. So as you can see on this graph on the top left of the screen, there's a significant increase in GLI1 and thereby hedgehog pathway activation in the P53RB knockout mouse osteosarcoma cell lines compared to the radiation-induced lines. And you can see across the graph next to it the individual responses in the individual cell lines. Now, what was also interesting is that when we co-treat these cells with the small molecule smoothing inhibitor sinanergib, also known as LDE225, and these are the red bars on this graph, you can see that it completely abrogates this hedgehog response. So this suggests that pathway activation is via canonical hedgehog signalling. So since canonical hedgehog signalling is also, canonical hedgehog signalling, which is smooth and dependent, is also dependent on the presence of this cellular organelle called the primary cilia, we wondered whether this hypersensitivity to ligand is simply due to an increase in the abundance of primary cilia. And so we're able to assess this by immunofluorescence using acetylated tubulin, which is a marker of the ciliary axonal microtubules, as well as ARL13B, which is a marker of the ciliary membranes. And you can see in these images on the right here that we can very clearly identify primary cilia on individual cells, and so therefore we can quantitate these. And so the bottom left-hand graph, you can see that there's a significant increase in primary cilia abundance in the P53RB knockout mouse osteosarcoma cell lines, again compared to the radiation-induced cell lines, and you can see the variability or the individual ciliary abundances in the panel in the graph next to it. Now, what you might note, and what's quite evident, is the variable responses in the CA45 lines in particular, and what's noticeable is that primary cilia abundance almost directly correlates to hedgehog ligand response. So if you look at CAOS25, so this cell line here, you can see that it's by far the most responsive of the radiation-induced cell lines, and it also has the highest abundance of primary cilia. And then if you look to the right of that, CAOS number 28, which is completely non-responsive and has no detectable cilia in that particular cell line. And this is quite consistent through the cell lines. I should note at this stage that we have sequenced P53 and RB in the radiation-induced cell lines, and there are some point mutations across the radiation-induced cell lines in P53, most of which have unknown significance, but there is a mutation in CA45 that results in a premature, CA4525, which is the responsive and ciliated cell line, which results in a truncation. So that's consistent with a potential role for P53 in eliciting this effect. And so we wanted to, I guess, show a more direct association between P53 and RB and hedgehog signalling and ciliogenesis. And so we re-expressed P53, RB, or both P53 and RB together in one of the highly responsive P53 RB knockout mouse osteosarcoma cell lines. And then we performed the same types of analyses that I've just showed you in the previous slide. So when we treat these cell lines with exogenous hedgehog ligand, you can see that there's a significant decrease in GLI1 mRNA, so a reduction in hedgehog pathway activation when we re-express P53, RB, or both together. And this, again, is associated with a significant decrease in primary ciliogenesis. So the abundance of primary cilia, as you can see in this graph, is much lower than that of the controls. And so this suggested that there was a direct association between P53, RB, ciliogenesis, and then ultimately hedgehog response. So if we look through the literature, there's some hints towards what the mechanism of this might be. And this is a really interesting paper from Pampliega and colleagues published in Nature nearly 10 years ago that really nicely showed that blockage of autophagy results in increased primary ciliogenesis and that these cells are then more responsive to hedgehog agonists. And they perform this using ATG5 knockout mouse embryonic fibroblasts. There's also quite a lot of literature demonstrating that P53 and RB can directly regulate genes that are required for autophagy. And then more recently, we've shown again in mouse embryonic fibroblasts that inactivation of P53, RB, or both P53 and RB together results in impaired autophagy as well as significant reductions in a large set of genes that are critical genes for autophagy that are known to be direct transcriptional targets of either P53 or E2F1. And so we hypothesise that perhaps this increase in ciliogenesis we see in the P53 RB knockout mouse osteosarcoma cell lines may be due to a reduction in autophagy. And so we assessed autophagic flux by looking at LC32, which is a marker of autophagosomes, using the lysosomal acidification inhibitor bafillamycin A1, or BAFF-A1, on this slide, in the presence or absence of serum. So in the presence of BAFF-A, autophagosomes accumulate, and the difference in LC32 in the BAFF-A treated cells compared to the control treated cells is indicative of lysosomal degradation or autophagic flux. Now, as you can see in the graph at the bottom, when we look at some of the radiation-induced cell lines that are non-responsive and unciliated, we see an increase in autophagic flux in the serum-starved conditions compared to the normal serum conditions, which is what is expected. However, we don't see this in the cell lines that are highly ciliated and responsive to ligand, and ultimately, when we look at these cell lines, have mutations in either P53 or RB. And so this is consistent with an impaired ability to induce autophagy, so an impairment of inducible autophagy. We've also looked at the potential effect of hedgehog signalling or requirement for hedgehog signalling in human osteosarcoma. And in the top of this particular slide, we've stained 120 osteosarcoma samples with 2 independent hedgehog antibodies. And we've been able to show that hedgehog ligand is expressed in about 50% of these patients. To be precise, it's 47% of the samples that we assessed. And it's associated with the high grade of disease and an undifferentiated pathology. And we're also able to detect the presence of a primary cilia in 70% of these samples. And so we've gone on and used a relatively small panel of human osteosarcoma cell lines, which at the time we had some limited genomic annotations, and we assessed these cell lines for primary ciliogenesis, just as we did before using immunofluorescence, and we were able to quantitate this. And really the take-home message from this particular slide is that in the cell lines U2OS and SJSA that are essentially very poorly ciliated, if cilia are detectable at all, tend to have intact P53 and RB. Whereas the ciliated cell lines, and there's obviously variable levels of ciliary abundance, tend to have some kind of genomic aberration in P53 or in the RB signalling pathway. And of course, we've also gone on to assess autophagy in this same panel of human osteosarcoma cell lines. And similar to what we saw in the mouse osteo lines, you can see that in the unciliated lines, we see an upregulation of autophagic flux, which is what we would expect. But this fails to happen in the ciliated cell lines, again, consistent with impaired inducible autophagy. Now, since smoothing is required for canonical hedgehog pathway activation, we wondered if inhibition or inactivation of smoothened in the p53-RB conditional mouse genetic model of osteosarcoma would affect the penetrance of the disease. And so the first thing that we did was to look at tumours from the p53-RB conditional knockout mice, and we're able to show that they do in fact express ligand, and they do have a high abundance of primary cilia, which is what we would have expected because that's what we saw in the cell lines derived from those particular tumours. Now, as expected, the p53-RB conditional knockout mice developed osteosarcoma with complete penetrance and a median survival of 137 days. In contrast, when we inactivated smoothened on the p53-RB conditional knockout background, we almost completely prevented osteosarcoma development. And instead, these mice developed these small calcified masses that predominantly occurred on the mandible and failed to progress. So in these x-rays here, you can see the p53-RB knockout mouse here has a tumour which is in the pelvis, and this is just the skull of one of the p53-RB smoothened knockout mice, and this is just the calcified mass here. And so they develop these masses quite early, and then they just stop and do nothing. And when we look at the histology of these, you can see that they're essentially entirely calcified. And these were defined as osteoid osteomas, or benign tumours of the bone. So this really indicated to us that while inactivation of p53 and RB is sufficient to initiate tumour development, these tumours tend to be dependent on hedgehog signalling in order to prevent terminal differentiation, as well as, I guess, for the propagation of this malignant phenotype. This was quite a profound result. We didn't expect to see something like this. So we wondered, I guess we wanted to explore the role of hedgehog signalling in more detail in normal osteoblast differentiation and bone development. And so during normal osteoblast differentiation, Indian hedgehog is the primary ligand, and it's normally secreted by the prehypertrophic chondroblasts. And it's absolutely required for the specification of these osteoblast progenitor cells from the osteoblast chondroblast precursors. So after these cells are specified, hedgehog signalling is no longer required, and it's rapidly downregulated, and is not needed whatsoever for the subsequent maturation steps in order to get a mature osteoblast or a bone-forming cell. And so we wondered what the effect would be, or the impact of sustained hedgehog signalling throughout this differentiation period. And so we utilised two distinct hedgehog gain-of-function models. The first is a constitutively active smoothened allele, the SMO-M2 allele, and the other is an overexpression of ligand using the sonic hedgehog transgenic mouse. And we crossed these mice to the osteoblast-specific Cree, the OSX Cree. And so you can see in these whole mount skeletal preps that are stained with alizarin red and alcium blue, these are embryonic day 18.5 embryos, but there's noticeable defects in skeletal development. And so these gain-of-function mice are really characterised by reduced ossification of the calvaria, the mandible and the maxilla, as well as reduced width of the long bones. And you can see an example of the femurs here. Now, when we take the hind limbs of the OSX Cree SMO-M2 embryos, as well as the OSX Cree as a comparison, what we're able to show is that there's a 30-fold increase in GLI1 mRNA. So that's, again, indicative of an increase in pathway activation. And we also see a significant decrease in these markers of various stages of osteoblast differentiation. So the pre-osteoblasts and terminal differentiation markers of mature osteoblasts. So we've lost, or we've got a downregulation of these markers here, suggesting that these cells are unable to terminally differentiate, and therefore we see this ossification defect. And lastly, we wanted to know what would happen if we inhibited the hedgehog signalling pathway in established osteosarcoma. So we've used a large panel of flank tumour models, both allograft and xenograft models. I'm just showing you three representative models on this particular slide. The one on the far left is a p53-RB knockout mouse osteosarcoma allograft model. So this is a highly ciliated cell line, which was quite responsive to hedgehog ligand in vitro. The middle panel is a ciliated human osteosarcoma xenograft model, and then the far right is a non-ciliated human osteosarcoma model. And you can see that when we treat these tumour-bearing mice with the small molecule smoothing inhibitor LDE225 or sunitinib, that there's a reduction in tumour growth and also a significant extension in survival. But it's restricted to the ciliated models. We see no effect whatsoever in the non-ciliated model. And when we look at the histology of these tumours at endpoint, we're able to see these focal areas of bone deposition. So these tumours actually had calcified bone within them, but it only occurred in the ciliated models treated with the small molecule smoothing inhibitor. It was not observed in the controls nor at all in the non-ciliated model. And you can see that really clearly with the alizarin red stained sections here, which is a marker of mineralisation. We've also had a look at some more specific osteoblast differentiation markers, and the top part of this panel is looking at immunohistochemistry for osteopontin, which is a marker of mature osteoblasts. So these are the bone-forming cells, and you can see that it's significantly upregulated in the sunitinib-treated ciliated xenograft and allograft models, but there's no change at all in the non-ciliated model. And at least in the allograft model, we see a significant reduction in RUNX2, which is a marker of the osteoblast precursor cells. So this is looking at the other end of osteoblast differentiation. So this really suggests that we're able to drive osteoblast differentiation by inhibiting the hedgehog signalling pathway. So we're working on this model currently whereby P53 and RB can regulate the expression of genes that are required for normal autophagy, that in turn mediates primary ciliogenesis and therefore cellular response to hedgehog ligand via the canonical signalling pathway. Our mouse models really nicely show that loss of both P53 and RB results in osteosarcoma but renders the malignant phenotype dependent on hedgehog signalling to maintain an undifferentiated state. And collectively we think that this implicates both P53 and RB mutation status as well as primary cilia frequency. I think that's important as potential biomarkers for hedgehog ligand sensitivity or for hedgehog ligand dependent tumours and therefore responsiveness to hedgehog inhibitor therapy. Now, some of this work has underpinned the development of a phase 2 clinical trial for recurrent adult sarcomas, and that clinical trial, using sunitinib, I should say, and that clinical trial is now finished, and we're currently undertaking this biomarker analysis in those samples, and we intend to correlate that with patient response on that trial. And I've still got a couple of minutes left, and so I just want to give you a little bit of an insight into maybe another direction that we've started to take this project into. And this is all very preliminary data and clearly got a lot of work to do, but I just would like to show you some of it just quickly. So I guess our redirection or evolution came about by the fact that in 2014, Mark Kieran published a really nice paper describing a phase 1/2 clinical trial for sunitinib in relapsed childhood solid tumours. And within that paper, he showed that sunitinib was well tolerated and did have efficacy, but only in those tumours that had a hedgehog activation signature. But one of the findings from that paper was that in a small number of children that received hedgehog inhibitor therapy, they actually had a premature closure of their epiphyseal growth plates in the long bones. So this was affecting their ultimate growth and development. And this is, I guess, it's an on-target effect of inhibiting the hedgehog signalling pathway in young children undergoing development. And so obviously this resulted in the need for caution when considering treating young children that are actively undergoing growth with hedgehog inhibitors. So we wondered if there was another way to potentially target these hedgehog responsive cells, and the biomarker for these cells is the primary cilia. And so we decided to utilise this wealth of data that has been alluded to in several talks today, at this conference actually, and that is utilising data that's been generated through the VPCC cancer model, Childhood Cancer Model Atlas. And in particular, the data that Christy Sun has generated, and Christy spoke a little bit about this in the flash talks yesterday and had a poster, so I'm not going to go into the detail, other than to summarise and say that Christy has extensively characterised 12 human osteosarcoma cell lines with high throughput drug screening as well as pooled CRISPR loss of function screening, and now we're layering in RNA-seq whole genome sequencing, methylation arrays and proteomics data. And all of that data you can see if you go to this portal and see what's available. So if I just concentrate on the functional data and specifically the CRISPR loss of function data, this is a limited or a custom pooled screen, so it's not whole genome. It's really comprised of 300 cancer-associated genes, and this is just a heat map summarising the data as it is. Below you can see a graph of ciliary abundance in the cell lines that we have the data for which we also have the loss of function CRISPR screening data, and we can arbitrarily bin them into ciliated or non-ciliated cohorts. And then we can reanalyse this functional data and see if we can determine any dependencies that might be specific for ciliated osteosarcoma cells. And you can see that in the heat map at the bottom of this graph. I hope you can appreciate that there are some differences. There's a few colour changes there that indicate there are some differences between the non-ciliated and ciliated groupings. And when we look at this in more detail, we're able to detect 42 cilia-specific functional dependencies in these osteosarcoma cohorts. And many of these targets seem to interact based on a string analysis. But what was really pleasing to see is that one of these targets was actually smoothened. So this was, we were really pleased to see this. Wasn't necessarily expected, but pleasing nonetheless. So when we look at the pathway analysis of these 42 specific targets, we can see that there's an enrichment for PI3K AKT signalling as well as epigenetic regulation. So these have been reasonably well studied in osteosarcoma and provide an avenue for further exploration in these ciliated cells versus unciliated cells. And then lastly, I just want to touch on some of the integration with some RNA-seq data that we have on those cell lines. So this is, again, early stages. We literally did this last week before we came up here. So rather than integrate the whole RNA-seq data across these cohorts, we've essentially just selected the 42 dependencies and then searched for their expression levels within the RNA-seq cohort. And you can see that we've graphed 3 of those as a representative example, being FGFR1, KMT2A, and PDGFRB. But you can see that the expression of those three genes is higher in the ciliated cohort than it is in the non-ciliated cohort or a normal human bone control. And in fact, all of these 3, as we know, there are pharmacological inhibitors that exist for these, so these are also avenues that we can explore along with some of the other targets. And then really quickly, in the last 30 seconds, we have begun to undertake in vivo pooled CRISPR screens, and we've performed this so far using 2 osteosarcoma xenograft models. So again, this is very preliminary. And it just so happens that one of them was a ciliated xenograft, one was a non-ciliated xenograft. And so when we interrogated that particular data looking for cilia-specific functional dependencies, we can further validate at least 2 of these targets, FGFR and KMT2A, as also being dependencies in the in vivo CRISPR screens. So with that, I'm going to stop. I need to acknowledge all the people that did this work, particularly Kathy and VJ for the Sonic Hedgehog work, Christy for the functional genomics work, as well as those involved in the VPCC. And I do want to highlight Carl Walkley, Maya Kansara, and Alejandro Sweet-Cadero for very generously providing some of the cell lines and animal strains that we used in this project, and also to Neil Watkins for his intellectual input into this project and our funding sources. And then just last of all, we have a new lab head at the Hudson who asked very nicely if I could just advertise the fact that she's looking for a PhD student to work on paediatric AML. So if you know of anyone who might be interested, please get in touch with Catherine. And I'm happy to take questions if we have time.
Thank you, Jason, for a very interesting talk. I guess now we've got maybe enough time for 1 or 2 questions.
Shout it out and I can repeat it if you like. Just shout it out. I'll repeat it into the mic for the virtual audience. Yeah, so that's exactly what we would predict. Yeah, so you can see in the in vivo study, for instance, there's no curative effect. So we do see terminal differentiation. So some of the cells do respond and will terminally differentiate, but not the entire population. Yeah, there's certainly variability, and obviously the higher responses come from those models that have a higher frequency of ciliogenesis.
Thanks, Jason. That's a great talk. Very interesting about the ciliated versus the non-ciliated. Marion Mateos from Sydney. My question actually is on the last bit of data that you started to show, showing the epigenetic inhibition potentially as a sort of pathway that could be targeted. And I was just wondering, given the challenge of using epigenetic drugs instead of in vitro and in vivo, how you were planning to sort of look at the readouts and interrogate that further.
Yeah, so we've done a little bit of work so if I put the cilia aside, we've done a little bit of work with HDAC inhibitors in osteosarcoma in the past, and we've shown that at low dose they're quite effective at differentiating osteosarcoma, and we see a similar terminal differentiation effect. So we can get mature bone in our animal models. That obviously progressed into the North trial, which is recently closed. The osteosarcoma cohort was, I think, ended well before the rest of the trial closed. I haven't seen all the data, but my understanding there wasn't any responders for the osteosarcoma cohort.
Jason, over here. Great talk. Really liked it. So this is a bit out there kind of question. So if you're inducing differentiation, but not effectively enough to result in long-term cures, so obviously you're not getting many of the cells, but cellular differentiation is also really intimately linked with cell cycle control, and you've deleted P53 and RB, so you've lost that cell cycle checkpoint.
Yep.
So then my question is, have you considered at least trying to restore the cell cycle checkpoints with an inhibitor like palbociclib, and maybe that would even push it even further once you can break the cell cycle, and then potentially then drive it even further?
Yeah, absolutely. So I haven't even spoken about the potential of combination therapy. Obviously, none of these therapies are going to work in isolation. That's certainly one possibility. The other thing we've considered is combining it just with standard of care. So I guess in other tumours that we've worked on, we've considered the fact that these primitive cells may be non-responsive to chemotherapy, for instance, but then if you can just nudge them out of their primitive state just a little bit, not all the way, not terminally differentiate them, just get them out of that initial state, they may become more sensitive to chemotherapy. So there's a few different strategies that we've considered. There's definitely a disconnect between the cell cycle and primary ciliogenesis in this particular case as well, which is difficult to comprehend. So normally the primary cilia would be retracted in S phase during the cell cycle, but we actually see primary cilia present during S phase within these osteosarcoma cells. So it's messed up.
Okay, so if everyone please join me to thank Jason for a wonderful presentation this afternoon.